Let us discuss different types of asexual reproduction. One of the most common method of asexual reproduction is binary fission. In case of binary fission, first of all, the nucleus get divided and then it is followed by division of cytoplasm. That is called cytokinesis. This binary fission, it is of different types. Simple binary fission, it occurs in case of amoeba. Longitudinal binary fission, it occurs in case of euglena, and transverse binary fission, it occurs in case of paramecium. How binary fission occurs in case of paramecium? This is paramecium. This is the cilia. Here is macronucleus. This is the micronucleus. This is the micronucleus. There is a contractile vacuole. Contractile vacuole. One contractile vacuole is present on posterior region also. This is the posterior contractile vacuole. This was the oral groove. This was the cytopharynx. As soon as transverse binary fission starts, first of all, oral groove degenerate, cytopharynx degenerate, macronucleus elongate, and later on micronucleus divide by mitotic division. This is the macronucleus elongated, macronucleus, and here micronucleus divides by a mitotic method. Then oral group develop in the posterior region. This is another oral group. Two oral groups. This macronucleus get elongated. Micro get divided into two, and then it is followed by constriction in the middle. constriction develop in the middle region. This is constriction, and now macronucleus get divided into two. Macronucleus get divided into two. And then contractile develop in the middle region. Contractile vacuole develop in the middle region. These are new contractile vacuole. New contractile vacuoles. Here is the macronucleus. Here is the micronucleus. Macro, micro, macro, micro, and two older contractile vacuole. This one is also older contractile vacuole, and then ultimately it get divided into two. And then oral group develop in the both regions. Cytopharynx develop. Cytopharynx develop. Now this is a complete view. This is the starting point: contractile vacuole, macronucleus, oral group, macronucleus, cytopharynx, and posterior contractile vacuole. Then after that, cytopharynx disappear, and then macronucleus start dividing by amitotic method. Macronucleus elongated, and then it is followed by development of two oral group, and then it is followed by constriction in the middle region, and then it is followed by development of two paramecia by developing two contractile vacuole in the middle region. Macronucleus get divided into two macronucleus. Micro also get divided into two, and then ultimately this paramecia get cleaved into two by formation of two cytopharynx also. Now let us see how longitudinal band fission occur in case of euglena. This one is the euglena. Here is the oral groove, cytopharynx. Here is flagella. Flagella 2 in number. This one is the larger flagella. This one is the smaller flagella. Here is contractile vacuole. Contractile vacuole. Here is eye spot. Here is the nucleus. What happens? This is euglena. It has chloroplast also. It takes part in photosynthesis as well as it may ingest food with the help of food vacuole. What happens during binary fission? Its chloroplast get disorganized and median furrow develop in the middle region in oral pharynx. Here is the middle ridge. Middle ridge develop. This is the median ridge. And then two flagella develop, two additional flagella. These are larger and these are smaller flagella. Nucleus of this euglena elongate. This one is the get it nucleus, and then a furrow develop in median ridge. This is a development of furrow in median ridge. This is longitudinal furrow. Here are flagella, larger flagella, smaller flagella. Two eye spot develop. Two contractile vacuole develop. This one is the eye spot. This one is the contractile vacuole. Here nucleus again elongate and get constricted in the middle. This is a constriction. In the middle region of nucleus. Gradually this longitudinal furrow elongates and nucleus finally get divided into two. Here are two flagella in each part, eyes part and contractile vacuole in each part. And then this is the furrow that develops more deep, and it causes this division of euglena part. Ultimately, euglena get divided into two parts. Here are two separate nucleus, contractile vacuole, flagella, eyes part, and then in this way we will get two daughter euglena by binary fission, and this binary fission is longitudinal. Once again, see this diagram. This was the euglena having larger flagella, smaller flagella, contractile vacuole, eyes part, nucleus and chloroplast. And then median furrow develop in the middle region and nucleus get elongated, and then a longitudinal furrow developed in the median ridge. Two eyes part develop, two contractile vacuole develop, two larger flagella, two smaller flagella develop, and constriction develop in the middle region of nucleus, and the furrow which develops is gradually moves in deeper area. Nucleus finally going to be divided into two parts by developing deep constriction in the middle region, and ultimately cytokinesis is completed and euglena is get divided into two daughter euglena. Multiple fission in amoeba. Multiple fission is a method of asexual reproduction by which large number of individuals are produced from single parent. Generally, it occurs during unfavorable condition, or it mostly occurs in parasites. Let us see multiple fission in case of amoeba. This amoeba has pseudopodia, and first of all, during unfavorable condition, pseudopodia are withdrawn, and then amoeba become somewhat spherical in shape, and it secrete a wall around it, That wall consists of three layers. This is a cyst wall. This cyst wall is secreted by this amoeba. This protect amoeba during unfavorable condition, This process is called encystment. Encystment and encystment is a mean for prenation. It is a mean for perination. And amoeba get preserved, and as soon as favorable condition returns, its nucleus start dividing into a number of daughter nuclei. These are daughter nuclei which are produced by repeated nuclear division. Repeated nuclear division, and due to repeated nuclear division, so many daughter nuclei are formed inside this cysted amoeba, and then each nucleus gets surrounded by cytoplasm. Cytoplasm get clips, and a small amount of cytoplasm gather around each nucleus. This is the small amount of cytoplasm. Amount of cytoplasm surrounds each nucleus, and then a wall is secreted. Wall is secreted by each structure, and these structures are now called as spores. These are the pseudospores. Pseudospores. And then as the favorable condition returns, this cyst get ruptured, and these pseudospores are liberated. These are the pseudospores which are liberated from this cyst. This whole process is also called as sporulation. This sporulation will also occur in case of parasite called Plasmodium. This is the sporozoite of Plasmodium, which enters into the body of human being as the female Anopheles mosquito pours saliva, and that saliva contains a number of sporozoites. In this way, sporozoites are inoculated into the human body, and they reaches into liver cell through blood circulation, and they enter into liver cell and get converted to a spherical structure. This spherical structure is called cryptozoite. Later on, what happens? These cryptozoates, they enlarge in size, they undergoes growth, and then each cryptozoite nucleus, this undergoes repeated nuclear division, and produce a number of daughter nuclei, and these daughter nuclei get surrounded by a small amount of cytoplasm. Cytoplasm get cleaved and a small amount of cytoplasm gather around each daughter nuclei, and in this way tiny structure is formed. This tiny structure is known as cryptomerozoites, and as soon this structure bursts, cryptozoites are burst, and these are cryptomerozoites are liberated into blood. These are the cryptomerozoite, and this structure is cryptozoite. They are liberated into blood. This whole process is known as multiple fission or zygogony. It will occur inside the liver of human being. This is a type of asexual reproduction in case of Plasmodium, which was a protozoan, a parasitic protozoan, which enters into the body of human by sporozoites. Sporozoites are the infectious stage. They enter into human body as female Anopheles mosquito bites a healthy person and she pours her saliva into human. They are present in saliva of female Anopheles mosquito. In this way, sporozoites get inoculated into human, and through blood circulation, ultimately they reaches up to liver cell, which is laden with nutrients. They attack liver cell. They become spherical in shape, called cryptozoite. Cryptozoite enlarges in size, and then its nucleus undergoes repeated nuclear division, and then it forms a number of daughter nuclei. These daughter nuclei get surrounded by cytoplasm, and each structure now develop into cryptomerozoite. And as the cryptozoite rupture, cryptomerozoites are liberated into blood. This is a multiple fission which occur in parasitic protozoans called Plasmodium vivax, which enter in human body as sporozoite.